In the previous video, we have seen the basic definition of a microscope and the different characters and parts of a microscope. In this particular video, we are going to see the types of microscope, that is light microscope and electron microscope, and the different subdivision that comes under this light and electron microscope. First, light microscope. What is a light microscope? So, as I already mentioned in the previous video, light microscope uses the properties of light to produce an enlarged image, and it's the one of the simplest type of microscopes. Under light microscopy, you have four types: bright field, dark field, fluorescent, and phase contrast. We will see one by one what are all the basic character differences between these four types of light microscope. The first type of microscope under light microscopy is bright field microscope. This bright field microscope is a basic standard instrument used in the laboratories to observe the microorganisms. It usually contains of two lenses, that is objective lens and the eyepiece lens, which we call it as an ocular lens. So this eyepiece lens or the ocular lens are the nearest to the eye, and it is further magnifies the images formed by the objective. So the second lens, that is the objective lens, are the one nearest to the specimen or the object, and it magnifies the object to a definite number of times. So you can see here in the image, this is the image taken using a bright field microscope. So the name of this bright field microscope has the meaning of what it does. So in the bright field microscope, the background or the background field will be bright and the objects will be looking dark. So this is how you can differentiate between this bright field and the next one, that is the dark field microscope. This dark field microscope is designed to eliminate the need for staining to achieve a contrast between the specimen and the background, because it uses a dark field condenser. It also contains an opaque disc which blocks the light that enters the objective directly. Apart from that, only the light that is reflected of the specimen enters the objective lens. It also has a condenser which directs the rays of light into the specimen field at such an angle that only the rays strictly an object in the field are bent or refracted into the objective. Most is like directly transmitted, while some is scattered from the sample, and only the scattered light goes on to produce image while the transmitted light is omitted. You can see here the image that is formed from this dark field microscope. So this is like completely opposite of what we have seen in the bright field microscope. Here the background or the field will be dark and the images will be bright. So the dark field microscope is particularly valuable for examination of unstained microorganisms, whereas in your bright field microorganisms you have to stain your samples. And here these unstained microorganisms are suspended in a fluid, wet mount, and a hanging drop operations. Now the third type of light microscopy is called as phase contrast microscope. This phase contrast microscope is useful because it permits detailed examination of internal structures in the living microscopes. So if you want to see an internal structure of a living microorganism, then you need to go for this phase contrast microscope. And it is like not necessary to fix or stain the specimen when you are performing under this phase contrast microscope. The basic principle behind this type of microscopy is based on the wave nature of the light rays. So what is this wave nature of the light rays? If the wave peak of a light rays from one source coincides with the wave peak of light rays from another source, then the rays interact to produce a reinforcement. One set of light rays usually directed from the light source, another set is from the light that is reflected or diffracted from the particular structure of the specimen. When these two sets of light rays are brought together, they form an image of the specimen on the ocular lens. It usually uses a special condenser that contains an annual diaphragm, or we call it as a ring. So this is the diagrammatic representation or a microscopic image that is obtained using the phase contrast microscope. The last type of a light microscopy is fluorescence microscope. So the name gives you the meaning what it does. Yes, it is designed to view the macromolecules labeled with fluorescent compound. It's basically used in hospitals and clinical laboratories to identify the disease-causing microbes. The fluorescent dyes are nothing but a chemical compound that is capable of absorbing the UV light and re-emitting the energy of visible light. The basic components in a fluorescent microscopy are a light source, an excitation filter, and mirror and a emission filter. So this is the image captured using a fluorescent microscopy. You can see with the help of the fluorescent dyes that's added to the specimen, the target areas which we want to highlight are fluorescent in nature, which we can easily observe and understand the components behind it. Fine. Now we have seen all the 4 types of light microscopy. Now it's time to move to the second category of microscopes, that is electron microscopes. So what's the basic thing behind this electron microscope which differs it from a light microscope? The electron microscope uses a beam of electrons rather than the visible light to illuminate the sample. They focus the electron beam using the electromagnetic coils instead of glass lenses. So these two are the basic difference between a light microscopy and an electron microscopy. And why they are using this electromagnetic coils instead of glass lens is because the electrons can't pass through the glass. The electron microscopes enable us to look in far more detail at objects than is possible with the light microscope. Some electron microscopes can detect the objects that are approximately 1/20th of a nanometer in size. They can be used to visualize objects as small as viruses, molecules, or even individual atoms, which can't be actually seen under a light microscope. Unlike the light microscopes, electron microscopes can't be used to look directly at a living things because of the special preparation that samples must undergo before they are visualized. This is one of the disadvantages that we have with this electron microscope, because we can't see a live specimen like how we can see in a light microscope. Instead, electron microscopes aim to provide a high resolution snapshot of a moment in time within a living tissue. So you can even record in detail of like every second movement of a particular living tissue. So this is a image that is captured using an electron microscope. You can see how minute and how clear even the smallest atoms are looking under this electron microscope. So under this electron microscope, you have 3 basic types. We can call it in short SEM, TEM and STEM. So what is the abbreviations of these 3? So SEM stands for Scanning Electron Microscope. So scanning electron microscopes lets us see the surface of 3-dimensional objects in very high resolution. It works by scanning the surface of an object with a focused beam of electrons and detecting these electrons that are reflected from and knocked off the sample surface. So this is scanning electron microscope. The second one is TEM. It's transmission electron microscope. It looks in very high resolution at a thin section of a sample, that is analogous to the compound light microscope. This makes it particularly good for learning about how components inside a cell such as organelles are structured. The third type combines both the scanning electron microscope and the transmission electron microscope. We call it as scanning and transmission electron microscope. A typical STEM is a conventional transmission electron microscope equipped with additional scanning coils, detectors and necessary circuitry, which allows it to switch between operating as a STEM or a CTEM. So I will now show you the different images captured using these 3 types of electron microscopes. The first image that you are seeing here is captured by a SEM, that is scanning electron microscope. You can see how the 3-dimensional image view are captured with the help of the scanning electron microscope. The second image is taken from TEM, that is transmission electron microscope. So even a virus or the internal structures or organelles of an organism can be observed under this transmission electron microscope. The last one is the basic concept behind the scanning and transmission electron microscope, which works on both the annular dark field and an annular bright field, giving a very clear depth of the organisms. So with that, we come to the end of this topic, microscopy.